Hello everyone, and welcome to this audio podcast. This audio podcast as part of the prompt series. This audio podcast is part of a ball and run ball. This audio podcast is part of an atomic headline, and this audio podcast as part of a Storm Robotics production. Through this audio podcast, some conclusions in around as part of this audio podcast series. So, um, Genomic amplification methodologies have provided it sort of a stimuli for assays based on DNA and RNA detection to be utilized or used in the form in the diagnostic in amongst the in amongst the laboratory diagnostics, and particularly in around virology, but also additionally in around microbiology and bacteria and to bacteriology or bacteria in the detection of bacteria. At best, assays are based on amplification of target or signal sequence for nucleic acid detection, provide a highly sensitive highly sensitive investigations for the diagnosis of varying sort of microbial and virological infections. Further, a modification of molecular assays is required or are required to allow to encourage to encourage and allow analysis of large numbers of clinical samples through through sorry large numbers of clinical samples, and where clinical samples, for example, are aligned to where investigations are through through the use of automation or automated laboratory investigation, but additionally with laboratory specialists and practitioners to determine varying microorganisms or viruses viruses, or through extraction through extraction and detection additionally. So that draws this audio podcast to a close. This audio podcast as part of the prompt series. This audio podcast as part of the ball and run ball. This audio podcast as part of an atomic headline, and this audio podcast as part of a Storm Robotics production.